For those of you who don't know me, and it's my absolute pleasure to introduce Leah Starita for today's Cancer Center Symposium. Little background about Leah. Leah is from New Jersey, went to Rutgers, as one does, then did her graduate work at Harvard, where she worked with Jeff Parvin, looking at BARD1, which is a key regulator of BRCA1. She then, as a postdoc, moved to University of Washington in the Fields Lab, where that's where we met. We were Baymates for a few years, became fast friends. Leah was working on high throughput methods for discovering E3 ubiquitin ligase substrates, and I was doing my thing. I left to come here, and in the meantime, Leah and several colleagues at the University of Washington developed techniques you probably know about, deep mutational scanning, which led to the Atlas of Variant Effects, which she'll discuss today. She's during the pandemic, she led what eventually became the Seattle flu study, and was one of the early pioneers in rapid public testing. They still test 500 samples a day, as I understand, something like that. So it's quite an effort to, now they're doing flu and all kinds of things. And I would say that sort of kicked off her career in managing extremely large projects, which she's continued on in the Atlas of Variant Effects, and has a large lab focused on understanding the potential functional impacts of variants of unknown significance, which is arguably the largest issue in human genetics today. So yeah, I think things that I love about Leah, she's irreverent, which I appreciate. She has a dogged determination to get to the bottom of something, which you'll see today, having started her career and continuing that on BRCA1. And she's just a wonderful person and speaker. And I have a little something for you. It is called, I don't know what it is. It is the Cancer Warrior Award from the Cancer Center. This is for you. And I will leave her to it. So welcome, Leah. Looking forward to your talk.
Notes, isn't it?
I mean, it's gonna have notes. Should I try to do this and this? And then
Go to PowerPoint.
yeah, this is PowerPoint. Go back to Zoom.
There's Zoom, I mean, yes.
Share.
Share. That's your notes page.
Right, but I can make it swap displays maybe.
Yeah.
Swap display.
Now they're seeing your notes in here.
Right. All right. You know what? Let's forget it and go back to the way you had it before. It's fine.
So you share screen again. Want me to?
Let's, or we can, you had it before, right?
Yeah. So basically, unplug.
You can see the notes on there. Yeah.
Okay, go ahead. And share your screen again. Back. You're gonna have to swap screen.
Okay.
Open PowerPoint. Go to swap screen.
All right, I can just end the slideshow. Just go here and then do this. And
You
then,
gotta open that. Okay, you're on.
okay.
Okay. And then you just advance it here. Click on the. Okay, there you go.
Okay. Well, thank you. Oh, yeah, I can do it the hide.
Slide floating.
There we go. Okay. All better, I think. Thank you very much. And thank you, Jay. I know Jay very well, like enough for him to have told something really embarrassing just about now. So I'm thankful for him for not doing that. And also Jay was the first person that really introduced me to bioinformatics and taught me that sort unique C sort minus NK is basically 99% of bioinformatics. And I really appreciate that. And I have taught that to an entire other generation of graduate students who also appreciate that Jay taught me that as well. So thank you very much. And thank you for that cancer warrior thing. I'm actually really, that's means a lot to me. Thank you. So I'm going to be talking today about understanding the effects of genetic variation at scale. I work for one of my appointments is at the Brotman Baty Institute for Precision Medicine, and kind of we're very, very good at being able to read the genome, right? Sequencing has won. We can sequence things now, we can sequence all of the things, but actually understanding what we're reading has become a massive barrier to kind of, I think what we all imagine with precision medicine is this like genome guided medicine where, you know, your precise sequence is guiding the treatment that you're going to get. And I'm going to use this little cartoon of BRCA1 to really bring that point home for understanding kind of what we need to know and the accuracy at which we need to know it. So this is a little cartoon of BRCA1. I've been working on this gene for a really long time. At its N terminus, it has a functional domain, and its C terminus, it's got another one. You need to basically make all of this protein in order for it to be a tumor suppressor. I'm just going to show kind of a little cartoon of three amino acids, three codons. And what happens when somebody gets tested for BRCA1 or for hereditary breast and ovarian cancer risk is that they can get a test back that says, you know, there's no problem, that they haven't found anything that's different. Or there can be a frame shift, so a single or double, something that takes the reading frame, and we'll have an early stop codon, usually leading to nonsense mediated decay and no protein. Same of a single nucleotide variant that causes a stop codon. And you can be pretty sure that these are going to be loss of function, and therefore you're going to lose the function of a tumor suppressor, and you're going to be at higher cancer risk. However, if you have a missense mutation, this is harder to understand, right? And the consequences here, if this is not pathogenic. Well, if it is pathogenic, this person would be offered risk-reducing surgery, which would then bring their risk from 50 to 70% lifetime risk back down to about 12, which is the rest of the world, or we'll do nothing at all, right, if it's benign. And so does anybody have any idea what this leucine to arginine change is going to do? Does anybody want to take a guess? Right, so guessing here is no good. You want to be really sure, and what happens is that a lot of times there's just not enough evidence to understand whether or not this is going to be linked to disease and therefore pathogenic, or not linked to disease and benign, and those become these variants of uncertain significance, these like question marks in clinical genetics, and they can't be used to guide treatment. And this is what the content of a clinical genetics database called ClinVar looks like for missense variants. More than 75%, about 80% of them are variants of uncertain significance. So of all the 2,000 genes or so for which there's variants in ClinVar, most of the missense variants are variants of uncertain significance. And I think this is probably pretty surprising to some people that these variants have just been found and there's like a collective, like, we don't know. Going on here, right? And this is actually it's a huge growing problem as well. So ClinVar was really started in 2015, and now variants were pathogenic or benign. And that's just not happening, because I think what is happening that if you think about the de novo mutation rate, right? So every person has 60 or 70 private mutations. If you think about the size of the human population, there's about 50 people walking around with every possible single nucleotide variant that is compatible with life. right? So I think what's happening is that as we're sequencing, we're finding those variants that are private to individuals and families, and there's never going to be enough evidence to really classify them. And what evidence do we use to figure out whether or not a variant is pathogenic or benign? We can use the patient phenotype. This works for really rare disease. It really doesn't work for things like breast and colon cancer, where the risk of finding somebody with breast cancer is not a surprise generally. You could do segregation analyses where you would sequence like the entire family and understand whether or not the variant is following the phenotype throughout generations. But if anybody has dealt with the US healthcare system, you will know that they're not paying for that. The other thing we can do is use population studies, right? So if you look at big databases like Nomad or the one generated by TopMed, and you can say, okay, well, this variant is occurring at too high of an allele frequency to be associated with that disease, right? The disease is only 1% and the variant is at 5%. That can't be causative. So that's really great for looking at benign variants, but this is also really biased, right? The populations that have been included in big genetic studies historically are very white, very Caucasian, very European. And so when we find variants from people in populations that have been historically underserved, those also become variants of uncertain significance at a much higher rate. And so not only do these populations have problems getting access to genetic medicine, but even once they get into genetic medicine, the likelihood that they get a non-answer is higher, which is really a shame. But there are two more other sources of evidence that you can use in variant interpretation, and one of them is predictive models. So things like alpha missense, things like that, although we can't use alpha missense yet because it's not been calibrated, I guess, for clinical use. And then there's functional assays, and these two things can be, you can imagine having a functional result or a prediction for every base change in the genome, right? And this is what we're going to talk about today, is trying to generate functional data in order to use it in clinical variant interpretation. And each one of these pieces of evidence can actually be used at 3 different levels, which we'll talk about a little bit later. So supporting, moderate, or strong. So going up in like how much weight they carry in variant interpretation workflows. And then, of course, how these pieces of evidence combine to get you into these classes. So, benign, likely benign, uncertain significance, likely pathogenic, and pathogenic. So, for example, you need 2 strong pieces of evidence to get to pathogenic, or 2 strongs to get to likely, things like that. Okay. So again, functional data can really fill that gap when this genetic data is missing. But historically, this has been done kind of one at a time. So somebody will have, populations of, BRCA1 variants, and they're like, oh, well, there's all these VUS, and we're going to test them kind of one at a time. I've actually done this. And what happens is, those variants are put in some database or something. It takes some poor graduate student or postdoc a couple of years to kind of look at them. And then, that information gets published. That information is usually generated too late for that person in which they found that variant, right? And also, I just told you that there are, 500,000 variants of uncertain significance in these genes that are linked to disease. That is a lot of graduate students and postdocs if we're only doing these 10 at a time, right? So to fix that, I've been working on generating or developing these assays called multiplex assays of variant effects, and to generate functional data for thousands of variants in a single experiment. And so the advent of DNA sequencing, or kind of the revolution in being able to sequence and write DNA, has enabled these technologies where we can take a library of genetic variants. So perhaps a library of all possible single nucleotide variants in a gene that you're really interested in. We can subject them to some sort of functional assay and mass. So for example, for a gene that is essential for cell growth, you can imagine that expressing a bad copy of this will make certain cells die and others could live. You can hook these things up to fluorescent reporter assays. MPRAs are of the same sort of assays. And then you can use this to generate what we call a variant effect map, where kind of before these variants are even discovered in the clinic, you've tested the functional effects of every possible single nucleotide variant or every codon swap, and you can, develop a map of what any one of these mutations would kind of do to the protein, whether it's now loss of function or not, or you've destroyed splicing or not, things like that. Okay, so I'm going to talk about a MAVE that we built for BRCA1. And so BRCA1, as you know, is a double-strand break repair protein. And we have this really nice haploid cell line. And BRCA1 and all of its friends in that pathway are essential in this line. And we're also going to make these mutations using CRISPR. So this haploid cell line is really handy for that, because when we go to edit, we only have to edit one copy of the genome. And so what we do is we're going to edit every possible single nucleotide variant into BRCA1, and then just wait to see which ones of those variants drop out of the population. Those are probably the loss of function variants, right? And so the way we do this, again, this is called saturation genome editing, is we express a guide RNA in Cas9 to an exon of BRCA1 to cause a double-strand break. But we have given those cells a library of repair templates. So using array technology, we make plasmids that have every single possible single nucleotide variant. And these are always in a pool. These are never separate, right? And so the cells are getting the double-strand break. They're getting these repair templates. And as the cell chooses one of these plasmids to repair with, this haploid cell line then incorporates one of these variants. Each cell then has a different variant after the DNA repair process is finished. Okay, and so we've got a population of edited cells, and again, this is a growth assay, so we're just kind of waiting. BRCA1 is essential, so we're just kind of waiting to see which of the variants cause enough damage to BRCA1 to start, to not allow the cells to grow anymore. And so, the trick to this is sequencing, right? So we sequence to identify and count each of the variants at these stages of this assay. So at day 5, and then again at day 11, and then we generate a functional score by measuring that depletion. Okay. We have to do this for every exon, or we have to do this for every exon that we want to target. So we can only target about 150 bases at a time. So we did 4 libraries to cover the ring domain and 9 to cover the BRCT, all of those exons. So this could have given us a possible scores for just about a little bit over 4,000 variants. And so we did generate those scores. So again, this is just the measure of the depletion of the variant from the population. It's a little bit more complicated than that, but not much. And so right around 0, we get the variants that didn't change. So it's a log 2-fold change of 0. And then at -2, we get this other population or so of variants that look like they might be loss of function. And if you look at the labels on some of these variants, you can see that the synonymous variants, the synonymous variants are around 0, except for about 1% of them that affect splicing. And then the nonsense and the canonical splice variants are here around -2. And then the missense variants are bimodally distributed kind of around those 2 populations, right? So one would suggest that maybe some of these missense variants that are equally as depleted as a nonsense variant is loss of function, whereas these other missense variants, about 80% of them overlap the synonymous variants, so those should be reference or wild type like function. But importantly, we did this in this crazy haploid cell line that used to be a chronic myelogenous leukemia. What does that have to do with breast cancer? right? So does this assay measure the same thing that is causing, does this assay for loss of function measure the same thing that is causing cancer in a human when BRCA1 might be loss of function, right? And so what we did was we took all the variants that were in the clinical genetics database that had already been classified as either pathogenic or benign, and we overlaid them on those functional scores, right? And these non-functional and functional tags are just those synonymous and nonsense distributions. And this assay really separates them very nicely, right? So all of the known benign variants, well, most of the known benign variants, there are some that are wrong, fall in the functional category, whereas the pathogenic ones fall in the non-functional or functionally abnormal category or loss of function, suggesting that, indeed we are measuring the same thing. These are the ones that are causing cancer in humans, and so this assay is able to separate that out. I don't think that would always be the case. So using all of this information, we're able to draw a variant effect map for these regions of BRCA1 that we targeted. So this is exon 2. All of these little rows are all of the position columns. All of these little columns are the positions in BRCA1, and all of the rows are the bases. This is pretty tiny, but the ones that are filled in are ones that are loss of function, and the ones that aren't are the ones that are functionally normal. And we're able to do that for that exon and all of the other 12 exons that we targeted. So if, for example, somebody was to walk into the clinic and ask, what is the function of this, 53T to A mutation, we would say, hey, that's functionally abnormal. Whereas if somebody asked about this other allele or this other variant, we would be able to tell them it was functionally normal. So this data, exists a priori before these variants are found by testing people in the clinic, which is pretty cool. Okay. So I told you that we're doing this to be able to reclassify VUS, right? I've just told you that we've developed this really cool party trick. Can we actually use this clinically to reclassify variants of uncertain significance? And in comes, this is a graduate student named Sean Fair, who's co-advised by myself and Doug Fowler. And he has a really interesting background. He is a certified genetic counselor and came back to graduate school and to do his PhD in genome sciences because he was tired of returning variants of uncertain significance to patients. Okay. And so this is kind of his mission to be able to understand VUS. And so what he did was he wanted to take all of this data and understand its clinical utility. And so about in 2019-20 or so, ClinGen, who is the clinical genetics arm, it's a big consortium under the NHGRI, developed a set of guidelines for how to use functional data, how to validate functional data for use in the clinical variant interpretation workflow. And what that is kind of a Bayesian framework for saying, okay, well, how well do the gold standard variants, so variants that have a known label on them as pathogenic or benign, how well do they separate in your assay and how many of them are there, right? And so for this assay, which separates them very well, and because it's BRCA1, there are a lot of known variants, we are able to use strong evidence. We're able to use this as strong evidence for and against pathogenicity, which holds a lot of weight. And so what we did was we had to collaborate with Ambry Genetics. So a lot of the other information that you need to classify variants is not publicly available in the United States, which is kind of a bummer, but Ambry Genetics is really great. I want to say that they've been super generous in sharing data with us, which is awesome. And so they shared all the data with us for all BRCA1 variants that overlapped our data set. So where we had data, and there were 110 VUS, or about a third of their database. And again, these are kind of the well-known areas of BRCA1. And then we applied the strong evidence for the loss of function variants for pathogenicity. We applied the strong evidence for the functionally normal variants as benign. And then we just use the ACMG interpretation framework that everybody uses from 2015. And we were able to reclassify 50% of the variants of uncertain significance, again, most of them moving toward benign. And this is really exciting to us that we were able to do this. So 50% in like one study is a really big deal. And so this is really exciting. And we actually used the companies will use a less conservative framework. And so, this was, conservatively 50%, which is pretty awesome. We were able to repeat this with published data for P10 and P53, and we reclassified 70% for P53 and 15% for P10. P10's got some weird problems where there are no known benign variants, so you can't really validate it very well. Anyway, but still, these are pretty good numbers. Another lab following the same kind of framework was able from Michigan, the Kitzman lab, they had a full set of variants for MSH2, which is one of the Lynch syndrome genes. And they were able to reclassify 70% of those variants. And then there's another group in England, we're able to reclassify 90% of the variants in GDX3X. This is really exciting to us. Ambry, when we did this exercise with them, they actually went and sent updated genetic test results to all the people that had gotten a VUS result in the past. So that was really exciting that we had kind of moved this technology. We kind of closed the loop. So we moved this technology all the way from the bench to the bedside in that sense. And so this works, right? We've got enabling technology. Got a problem. We want no more variants of uncertain significance, especially missense variants. So how do we get there, right? What is the vision? So the first is scale. So we have these assays. We just need to kind of turn the crank. And so we're able to do this kind of through the impact of genetic variation on function consortium through the NHGRI. So we have these two assays, the one I told you about already, which is cell survival and saturation genome editing. And another assay called protein abundance or variant abundance by massively parallel sequencing measures protein abundance. And these two assays are kind of generalizable over a lot of genes. So basically anything that is essential in the HAP1 line, or about 50% of genes work in this protein stability assay for reasons sometimes we understand and sometimes we don't. And the variant abundance assay is super easy. We just fuse the gene to GFP and look for the effects of missense variants on the stability of that protein fusion, right? A lot of the time when you have a pathogenic variant, it causes the protein to unfold, both that protein and the GFP get degraded. And we're able to really use FACS and sequencing to separate those variants. And so through this consortium, we're applying these technologies to a lot of different genes. We spent a lot of time optimizing these. So we want to be able to get much cleaner data. We want to be able to do it faster. So for example, this is a PALB2 data set that's got 10,000 single nucleotide variants. And then again, one of the hardest parts of doing this is actually getting one and only one variant into these cells. So we spent a lot of time making new landing pads and things like that. And as I told you before, every 150 bases needs a new library and a new guide RNA. So we have to do a lot of cloning to do this. And so we've been adapting a lot of those cloning steps to automation. This is a little mantis, and we're doing 96-well transformations and stuff like that. So it's been fun to do that process engineering. And then the genes we choose to target are chosen for a lot of different reasons, but mostly clinical utility. So the ones that are marked as ClinGen, so these are the ones that are on a special list of genes called the ACMG78. And these are usually adult onset diseases that can be, they're medically actionable, right? So if somebody is found with a pathogenic variant in one of these genes, you can intervene to reduce morbidity and mortality. The ones that are marked in Gregor, I collaborate with a rare disease consortium. So a lot of these genes are ones that would support novel gene discovery for Mendelian disease or phenotypic expansion. Some of these are just hereditary breast and ovarian cancer because that's where my interest lies. And then CPIC is the the Pharmacogenetics Consortium, trying to understand whether or not mutations in proteins that metabolize drugs, what they do, and try to understand how a person might be affected by different drugs. So that's how we choose these genes, and we are supposed to generate 200,000 variant effect scores over the next couple of years, which is really exciting. So we have scale. The next thing is new technology, right? So a lot of the genes I've talked to you about today are genes that can be expressed and assessed in random old cancer cell lines. But that is not the case for a lot of genes. So what we're trying to do is move some of these systems into differentiable cell types like induced pluripotent stem cells. And then the idea would be to edit the stem cell, differentiate into different types of cells, and then have different phenotypic readouts for each of these cell types. So for example, you could use cell survival, something based on flow cytometry, or even single cell analysis in the end. And so we chose as an example here a gene called MYBPC3, and it underlies hypertrophic cardiomyopathy risk. It's myosin binding protein C3. It's a component of the sarcomere, and it is only expressed in cardiomyocytes. Okay, and it's only obviously functional in cardiomyocytes. So it's not something we can assess in these kind of utilitarian cell types. And so, as I told you, it's part of the sarcomere, and it connects the actin to myosin. It's this kind of yellow snake, and this C10 domain is where we targeted to make these mutations. It's a very big protein. The C10 domain is a hotspot for pathogenic variants, and we also know that pathogenic variants in C10 in the C10 domain cause the protein to mislocalize and to become degraded. So we can kind of use that as a phenotype. And so human cells are annoyingly diploid. It's kind of a bummer. So how do we deal with that? So hereditary HCM is autosomal dominant. So we want to make one mutation on one allele. So the way we deal with that is we make a line that replaces the last 5 exons and introns in this gene fused to GFP. But importantly, what that also does is it knocks out a guide site so that when we go in to put the allele, the variant library in, it can only go onto one allele. And so we then target the other allele with a different color. So this has M-apple on it. And in this case, this is exon 32, and there are 500 variants in that library, making every possible single nucleotide variant in exon 32 plus the intronic regions around it. So then we have these edited pluripotent stem cells. And again, we're really worried about scale here. So in the past, people have only made, libraries of like up to 10 things in stem cells. So this was worrisome that we were trying to get 500 things in there. And then we take that edited pluripotent stem cell population and then differentiate them into cardiomyocytes and look at the expression of both of these alleles. So what does this end up looking like? So we have an edited iPSC. We differentiate those into cardiomyocytes, which is about a 2 to 3 week differentiation. And then we've got that wild type allele tagged with GFP, and you can see the nice sarcomere localization there. And then the variant alleles are targeted with M-apple, and you can see one that looks very similar to the GFP, suggesting is probably normal function, and then one that's just gone. And so maybe that's a variant that causes mislocalization. And so how are we going to assess this in mass? So we have these differentiated cardiomyocytes that have all these different alleles in them. They've got a bunch of different range of red fluorescence. We can sort those by the red fluorescence over the green channel, and then we can generate a score, right? So maybe variants in bin 4, we just use a weighted average of the variant counts in each of those bins. And so a high score means the variant had a lot of brightness, and a low score means that the variant was dim. And importantly, again, we tried to get 499 variants in, and we generated scores for 493. So that was the first thing we were totally psyched about, because again, we were very worried about the scale here. And you can see that they're quasi-bimodal, which was pretty awesome. And then if you color these by whether or not by their functional consequence, you get the stop codons at low scores, which is good. The synonymous codons are kind of spread across the normal range, but the missense variants go kind of across the whole range, which is really exciting. And some of these were in the C10 domain, and some of them were kind of upstream of that. And all of the C10 domain ones are ones that are depleted, which is exciting. And most importantly, there are variants of known effect in ClinVar that are in this domain and in this exon that we tested. And this assay does a really good job so far of separating those pathogenic and benign variants, which is pretty exciting. So even just this one exon alone gets us to moderate evidence for these variants, which is great. And so, we plan on for this assay. This is one of the slides. This is one of my bad decision slides that I took out on the airplane on the way here. So what we want to do with this is we want to target the rest of the exons in C10 with MYBPC3. And then we have a couple of other genes that will show that we're going to target that one that's in causes a neuronal phenotype, and one that causes an erythrocyte phenotype, to be able to assess whether or not this is going to work across we can edit in the stem cell, but how good are we at assessing these effects in these differentiated cell types, which is exciting. Okay. So, we like to tell the NIH that we can solve this ourselves, and we do say that when we write grants, but that we know that that is not true. And so the next thing we want to do is kind of galvanize the community to generate this kind of data to inform clinical genetics. And so Doug Fowler and I, along with some other like-minded people, started the Atlas of Variant Effects Alliance. And the goal of the Atlas of Variant Effects Alliance is to build kind of what is depicted in this cartoon here. So an atlas is a series of maps. So you can imagine, variant effect maps for all proteins or a lot of proteins that are associated with disease, and having those maps available before variants are found in these genes. And so the goal is to kind of generate the infrastructure and the standards and resources in order to kind of democratize these technologies and collect the knowledge of all the people that are out there in that international community. So it was founded just 3 years ago. It already has 500 members from 105 institutions in 30 countries, which is really exciting. We host the annual mutational scanning symposium, which was in Seattle, but went to Toronto and then Hingston, and we'll come back to the Broad actually. We host a variant effects seminar series that is led by trainees and for trainees and early career PIs. And then we do our work in 4 interconnected work streams. And what we're trying to build right now is a lot of kind of standards and minimal information standards, things like that, things that are kind of really missing in the field, but would be really helpful to be able to compare data sets across each other, be able to publish data sets and have somebody, know what you did, things like that. So we're working there. We also build tools for the community, and one of them is MaveDB, which holds all of the data that is generated by these assays. And this is really a baby of Doug Fowler and Alan Rubin. Alan Rubin's at WeHi in Melbourne. And right now, MaveDB is holding about 10 million variants, which is great because that's, it's bigger than ClinVar now, which is really exciting. And it keeps growing. It keeps growing. It's a little bit hard to use at the moment, and we're trying to fix that. Okay. And then the next thing is clinical outreach. So I told you we're generating all of this data, and it turns out that clinicians will not look in supplementary table 2 for the data. And so they just send us emails. And so the way this data is basically getting into the clinic now is they email me and I tell them, I give them the up or down, and then they do what they want with that data. And that is not good. And so one of the places that clinicians always go, like their first stop is always ClinVar, right? And ClinVar now will take functional data. They give you one sentence that is, and you can see that it's, to describe the assay, that it is all sorts, it's got great formatting problems. And I'm not sure how you're supposed to really have somebody understand what the assay was and what that score means in one sentence in ClinVar, right? Because it does say functionally abnormal, so that's good. But what does a score of like minus 1.98 mean? Who the hell knows, right? So we're working with ClinVar first to fix their data model because it's a mess. And second, to be able to federate across MaveDB and ClinVar so we can feed variants into them. And then clinicians can come back to a page that will kind of show them the distribution of all the scores and where their variant is. So we're pretty excited about that. And I don't know. Okay. So that's exciting. And then it turns out that most of the time when you talk to a clinical genetics professional, we're talking about like right now, the fellows that come into the University of Washington, and you say, oh, have you looked at the functional data? And they look at you and they say, what? And so the next thing we're trying to do is try to learn from our local, we've got one of the oldest and largest clinical genetics departments in the country, but there's no kind of effort to move this functional data that we're trying, the department next door is trying so hard to generate kind of into the clinic. So we've got this collaboration where we're trying to understand how we use it, how they use it locally in order to build a series of workshops where we kind of teach people what it means, we teach people how to read it, and we teach people how to use it in these variant interpretation frameworks. So we're really excited about that, and we'll be attending meetings like ACMG and ASHG and holding seminars there. Nice. Okay, so this is the vision. So we're trying to go from, again, enabling technologies all the way to no more variants of uncertain significance. And we think through all of these kind of integrated efforts that we should be able to get there. Maybe not in the next 10 years, but we should be able to make like I think, a really big push, especially into genes that we know are super important to the clinical genetics community, at least within like the next 5 years. So hopefully we'll get there. We're excited to do that. So this is our teams. Again, this is the mutant bats. I have a bunch of different, I run 3 different labs. So this is the team who did a lot of the work here. And then again, you saw the work of Sean Fayer, who did that iPSC work and the clinical validation work. And with that, I'd like to thank you and take questions. Yeah.
Stuff. So just a quick question.
Mhm.
It's probably an obvious one. Have you thought about extending this to somatic variants?
Yes, there are folks at the Broad. Mostly, there's been a pretty decent effort at the Broad, so I've tried to stay away from it because the Broad will crush you. So yes, so there has been, I know a bunch of different labs that are working on this from like that perspective, and also resistance to primary and secondary inhibitors of those kinases as well, to try to learn what's going on and be able, hopefully, to be able to design better kinase inhibitors that will overcome some of those mutations as well. Yeah.
That was really cool. So
Thanks.
I wondered about in your haploid cell line, when you're doing this, if like with the mutations that there are the variants that don't cause or cause very little trouble in BRCA, say, for instance, whether so it's possible because it's a haploid that they're sort of they're suppressed. And I'm just wondering if you would ever or have thought about doing a co-analysis with variants in, I don't know, genes that are known to, there might be regions of the protein that are more easily suppressible by second sites than others.
Yes.
It's like really powerful to do these haploid.
Yeah, but as you said, it's limited in a lot of ways. Like, where we draw that cutoff for where there's function and no function, it's not arbitrary. It's whether or not it overlaps the stop codon significantly. So in that case, it's not arbitrary, but it is because it has nothing to do with whether or not there's disease risk. We have shown that if you look at biobank, if you look in like the UK biobank and look at the ones that we called functionally abnormal, there's an odds ratio of 10, but if you look at the ones that we call functionally intermediate, there's an odds ratio of 3. So there's signal in there. So could we increase that signal by, you can imagine treating the cells with a DNA damaging agent and making BRCA1 even more essential. Or one of the things we would really like to do is understanding, so this is all in a vacuum, right? I would love to understand more about the genetic background that these cells are working in, because we know, for example, that there's very big differences in penetrance based on polygenic risk of these people who are carriers. It can go all the way from 20% to 100%, right? So what are these permissive backgrounds and what are these backgrounds where BRCA1 is more essential? And even in a lot of these proteins, there are missense variants that are at a high allele frequency. For example, there's a great one in P53 that it's at 50% frequency, and there's hundreds of papers showing that it is not really normal. And so I would love to do these same sorts of assays on both of those backgrounds, but these are kind of pretty hard. And so generally in the first pass, we just do the normal background. So yeah, exactly. The vanilla assay, if you will. Srinivas?
Yeah, so for clarification, so you said for the BRCA, you would expect 50% from the clinical database, the US. The other 50% were all scored in the middle in your MAP score. So would those be pushed by your bacteria damaging agent?
That is a really good question. I don't know, but then I would also not know. Yeah.
Might have a tendency to go low penetrance for
Right. One of the things we don't know is whether or not those things are in the middle due to technical noise. So one of the things we really want to be doing is doing a lot more replicates to really understand that, what that variant score is and its kind of confidence interval that score is actually supposed to be there and not in the middle because of technical noise. So we need to get our shop in order to be able to do that. And the other thing is, most of those variants that were unable to be assigned had a functional score, either pathogenic or toward pathogenic or benign. A lot of it is just all of the other evidence that is missing, right? So you can't take a functional score and get to pathogenic or benign or likely pathogenic or likely benign. You need more evidence. And when that doesn't exist, and so that's where most of that is missing, actually. Go ahead.
Where there's some version of that, so they don't get anemia, but later on in life they still get cancer. So the question is, could you somehow use your system to predict? Because the only ones they're looking at are the ones that do get cancer, those are the ones that present. So the question is, could you look at that, be able to predict where those people would be high risk?
Probably. Yeah, but is it because the reversion, there's still some cells left that have the mutation?
Still somewhat functional.
Oh, I see. Right. So how much residual function do you need? And can you compare that to the people who have gotten cancer? Yeah. Yeah, you'd have to have enough examples of that to know kind of where to draw that functional cutoff. And I don't know. Go ahead, Jay. Ray, yeah, Ray on it. Yep.
part, what are the point?
Yeah.
Fire. Habituating the effect.
Well, yeah, we do multiple replicas. And luckily these are human cells do not grow as fast as yeast. I would definitely expect second site suppressors if I did this in yeast. But we don't, I mean, they only 13 days is really only 13 generations. So I'm not too worried about that in these human lines. And you know, we make each mutation. Hopefully there are thousands of thousands of different edits. So hope, you know, if that does occur, hopefully it's random. But because we're asked that all the time, like, what about off-target editing? I'm like, well, it doesn't seem to be a problem. Okay. Well, thank you very much.